Welcome dear students, welcome to your tutorial channel Brilliant Bios. In this class we are going to learn about another cell signaling pathway that is called Hedgehog Pathway. At first I will give you some basic fact about this Hedgehog Pathway so that you can understand this pathway easily, then I will go into details. The first thing you have to understand the name of this pathway, hedgehog. This is very interesting because hedgehog are some species of spiny mammals. So why the scientists have given the name of these animals to this pathway? There is a reason. The reason behind this is the discovery of this protein, hedgehog. Scientists have discovered this protein in the Drosophila species. They found that loss of function of this hedgehog protein causes the fruit fly larva to be covered with pointy denticles, or we can say bristles or hair-like structures that resembles the spines of this hedgehog species. So from that discovery they named this pathway hedgehog pathway. Now the basic fact about this protein are like this protein hedgehog actually acts as a morphogen. I have already discussed what is morphogen, how this morphogen functions. I will also discuss about this in this class. That is, let's suppose this is a cluster of cells. These are all cells. Those actually secrete this molecule. Now this is called source from which the cells are secreting this hedgehog protein. These cells are secreting hedgehog protein. Now you have to remember this, that this morphogen do not actually go very far away. They remain very close from the source. These morphogen molecules, let's suppose this is hedgehog molecules, remains very close from the source. But there is a difference. That is, the concentration of this morphogen determines the fate of the cell. Like those cells are present very close to the source. They receive high concentration of this hedgehog. And those cells are distant from the source. Let's suppose these are cells that are distant from the source. They receive less amount of hedgehog proteins. And this concentration, not the molecules, because these cells also receiving hedgehog, these cells also receiving hedgehog. But as the concentration differs, they develop different cell fate during the development. So this hedgehog protein also play important role during the development. You have to understand this development, like lung morphogenesis, limb patterning, nerve system development, hair follicles development, spermatogenesis, bone formations, gut formations. There are so many organs. Actually scientists have found that in case of vertebrates, all the organs during their development require the hedgehog pathway. So this is very important pathway. Okay, now I will give you some example of this hedgehog protein that is homolog to the Drosophila, because scientists have searched this hedgehog protein to other organisms like vertebrates. They found three homologs, at least three homologs are present in all vertebrates. They call it sonic hedgehog. This name also is very interesting, sonic, I will tell you. Then another they call Indian hedgehog. This Indian hedgehog is a species, living species, and they found another protein, they name it desert hedgehog. So do you know that these two hedgehog name or protein name actually names after the two living species of hedgehog. They are two mammals, Indian hedgehog and desert hedgehog. But the sonic hedgehog is actually a cartoon character, Saga Genesis. Okay, interesting fact. Now there are some functions, particular functions of this hedgehog. Okay, and among them the major important functions are played by the sonic hedgehog. They perform versatile functions. The major important function I want to mention here, that is the limb patterning. So it actually helps in the limb patterning. What happens, I will not go into details because I will make a separate video on limb generation in the development biology class. So what happens during the development of limb, a limb bud generates. This is called limb bud. Limb bud. Do you know that our pinky, the smallest finger, is always the posterior of our hand? This is called pinky. This is always the posterior portion of this hand. So the sonic hedgehog plays this role. what happens during the development of limb bud, this sonic hedgehog, this protein is always also present in the only present in the posterior segment of this limb bud. Let's suppose this is the posterior and this is the anterior portion. No sonic hedgehog present in the anterior portion in normal development, during the normal development. But if you artificially, or if there is any problem, then if the sonic hedgehog starts to express in the anterior portions, then a phenomenon occurs that is called polydactyly. That means extra digits, formation of extra digits. So there are so many roles that is present by this, like the desert hedgehog actually present in the Sertoli cells, and if there is any problem of this desert hedgehog G protein, then there is hampering in the spermatogenesis process. Then the Indian hedgehog is actually helps in the gut and cartilage formations or bone formations. Okay, cartilage. So these are all important facts. Another important facts I want to mention here, that in case of mammals, a very interesting structure is actually found in all mammalian cells. It is called primary cilia. You know, primary cilia. This is not actually conventional cilia. We know that cilia, flagella actually a motility structure, but this primary cilia is very bizarre actually in function and also structure, because in case of cilia and flagella, the common structure is 9+2 microtubules, 9 outer microtubules and 2 core microtubules are present. But in case of primary cilia, only 9+0, that means there is no center microtubules are there. And this primary cilia, scientists actually work on this primary cilia. It is very interesting organelles in the mammals. They found that this primary cilia is the only region where this hedgehog pathway occurs, primary cilia, in case of mammals. Okay, so now I will go to the cell signaling pathway. What happens? Let's suppose this is a plasma membrane, and the receptor for this hedgehog protein is actually another thing is that the very important point I have just forget to mention, that this hedgehog protein actually go through a processing mechanism, because during the generation of this protein, it actually produced as a precursor protein, precursor. That means there is a 2 fragment of this protein. Let's suppose this is a 20 kilodalton protein, and there is another domain of this protein called 25 kilodalton protein. Let's suppose this is the N-terminal segment and this is the C-terminal segment. This hedgehog protein have autoproteolytic capacity. That means it can cleave itself. It can cleave in these portions, and the C-terminal fragment of this protein actually gets degraded. This portion is actually degraded out. So this is not functional portions. So I can remove this. Now this N fragment of this protein actually gets modified 2 times. First modification is the addition of a cholesterol at the C terminus. So here a cholesterol add, and this addition of cholesterol is very important because this addition actually helps in the binding with the receptor. If there is no cholesterol present in the hedgehog protein, it cannot bind with the receptor. Now another important modification occurs in the N terminus, that is the addition of a fatty acid called palmitic acid or palmitoyl group. Both these lipid modifications make this protein more hydrophobic in nature, so that it will not diffuse so far from the source. So this is another important fact. Okay, now I will go to the cell signaling pathway. Let's suppose this is a plasma membrane. I will show you two because here I will show the negative where there is no hedgehog protein, and in the presence of hedgehog proteins, 2 phenomena occur. In case of hedgehog protein, the receptor of this protein is called patched. The protein that actually receives this hedgehog protein is called patched. This patch protein is actually 12 transmembrane spanning protein, integral membrane protein. 12 membrane spanning region are there. Membrane spanning alpha helix are present. Okay. This receptor actually inhibit a protein that is present in the vesicles. Let's suppose this is a vesicular structure. This protein is present in the vesicles. Let's suppose this is another protein that actually inhibits the protein. This protein is named called smoothened. Okay, so patched protein inhibits smoothened, that present in the vesicle, that is present in the vesicles. And how this patched protein actually inhibits smoothened, it is not discovered till date. And this patched protein inhibits and causes the degradation of the smoothened, degradation. And how this patch protein actually causes this degradation of the smoothened, it is also not discovered. But scientists have found if you mutate this protein, if you mutate this patch protein, the phenomena occurs that looks very similar, like the embryo is actually flooding through the hedgehog proteins. I am telling you another time. If you want to know a function of a protein, you have to mutate the protein. You have to mutate the function of the protein. Scientists when mutate this patched protein, when they mutate this protein so that it cannot perform its functions, they found that it looks very similar, that the organism is actually flooding through hedgehog protein. That means huge amount of hedgehog protein is present. It behaves like that, but that is not the case. So that means this protein is actually inhibitory function. If you inhibit this protein, then it cannot inhibit the pathway. So the hedgehog pathway go through in normal situation by a negative feedback mechanism, that means patch protein and negative inhibitory protein inhibits the smoothened protein. Okay, now the smoothened protein has a very interesting fact, that is the smoothened protein in the C terminus, there is a tail-like structure that actually when it is present in the vesicles actually form a loop-like structure. It is not open in conformations. Another important fact now I will discuss about this. This is the main function, main part of this pathway, that is a multi-protein complex actually perform its function during this hedgehog pathway. I will give you the name. Let's suppose this is a protein. This protein is called fused. This is actually a serine threonine kinase enzyme. There are multi-protein complex. Let's suppose another protein. This protein, they actually associate with each other. Maybe this protein, so this is the fused, maybe this is the CK1 casein kinase enzyme. This is also a kinase enzyme. There are so many kinase enzymes are present. Let's suppose there is another kinase enzyme, you can call it GSK 3 beta, that means glycogen synthase kinase 3 beta. There is another kinase enzyme that is called pKa. Let's suppose this is a pKa enzyme. Not only that, there are so many proteins are also involved. There are microtubules, and this microtubule associated protein, a protein is present we call costal 2. It actually functions like the kinesin like protein. So let's suppose this is the costal 2, this microtubule associated protein. There is another protein that is associated with this costal 2, we call it SUFU. So this is a multi-protein complex, SUFU. And the ultimate protein that perform its functions is the another small protein called CI. In case of Drosophila, I am telling you this is a Drosophila pathway. The homologue of the CI protein, CI means cubitus interruptus. Cubitus interruptus. This is the protein that present in the Drosophila. The homologue of this protein in mammals or vertebrates, you can call it GLI. The homologue of the protein functions GLI. Okay. Now, another thing is in case of vertebrate, the costal 2, the microtubule associated protein, the homologue of this protein that is present in the vertebrate, we call KIF7. This is two information. Rest of the proteins are actually present in both Drosophila or the vertebrate. Okay, what happens actually when there is no hedgehog proteins? Patch proteins inhibit smoothened remains in the vesicles and smoothen go through degradation process. How this occurs, we don't know. But the fact is here, this GSK3, this three or four, you can say, kinase like GSK3, PKA, CK1, and this fuse, these all proteins actually phosphorylate the CI or cubitus interruptus protein, phosphorylate this protein. And when the CI protein gets phosphorylated, it actually targeted by a ubiquitin ligase enzyme that actually that will actually go through the proteasomal degradations, and ultimately the CI proteins actually gets fragmented, and the fragment of this CI protein called CI75. Now the CI75 is very interesting because this is actually a transcription factor, zinc finger containing transcription factor. Now the CI75, that is the fragment of the CI. This is the fragment. Let's draw me in this red color. Let's suppose the CI is actually the fragment of this CI proteins. It enters into the nucleus and it actually sit on the DNA that is a target of this Hedgehog pathway. And the main important function of this pathway is that CI75, the clip portion of the CI, actually inhibit these genes. Now you can see in the absence of Hedgehog protein, the target gene of Hedgehog pathway is actually inhibited by a very complex proteins. Now in the presence of Hedgehog, the patched proteins, when associate with the Hedgehog, let's suppose this is the Hedgehog, when Hedgehog protein binds with the patch. Another information I want to mention, that smoothen is actually a 7-membrane spanning alpha helix protein, integral membrane protein, 7-membrane spanning protein. When Hedgehog bind with the patch, a receptor mediated endocytosis occur. That means this the patched and the Hedgehog get internalized, or you can say endocytosed and get degraded. They actually get degraded. This is the mechanism. Now the inhibitor is actually get degraded, or no, there is no inhibitor. In that case, the smoothened protein actually go from these vesicles to the plasma membrane. Let's suppose now this is the smoothened protein, SMO in short. I have mentioned a very interesting fact that the smoothened have a C-terminus loop-like structure. Now it is open in conformations, and when this smoothened C-terminus C segment actually remains open in conformation, then this kinase enzyme like CK1, PKA and GSK3, these three kinase, CK1, GSK3 beta and the PKA, these three phosphate kinase phosphorylate the C segment of this smoothened protein. When this protein gets phosphorylated, this fused protein and the costal protein, you have to remember this fused protein, let's suppose it is a fused protein, this is also a kinase enzyme, serine kinase, and the costal protein, that is actually a microtubule associated protein, get attached with the C terminus of this protein, let's say costal 2. When these two proteins get associated with the C terminus, they also get phosphorylated. And the very interesting fact is, when this cos2 and fused bound with this C segment of the smoothened, then this multi-protein complex no longer remains. Then the CI, that means the cubitus interruptus, actually released, and it is not get phosphorylated by these kinases, and the total CI then enters in the nucleus and it sit on this DNA CI with a associated protein called CREB protein, CREBP, CREB, okay, CREB protein associated with this protein, and now it actually promote the transcription of the target gene, promote the transcription of the target gene. That is how the hedgehog protein pathway functions. But in case of mammals, there is a primary cilia that actually where the cell segregation actually take place. So if you want to know about this pathway more details or any topic, you have to join my live class. So for this video, this is the end of my hedgehog pathway. Thank you for watching and always keep learning.